Hi everyone, my name is Nick Mohrdieck. I am a senior at the University of Wisconsin and I am studying genetics and genomics. And so one of the classes that I had to take as a genetics major was a virology class where we happened to actually learn about coronavirus. So I'm hoping today that I can maybe give a little bit more insight into what the coronavirus is, especially since it's basically engulfing our world right now. And I want to share a little bit about some of the good news on coronavirus, especially on the vaccine front, and potentially teach about what that vaccine strategy is and how it can actually help us become immune to the virus and potentially stop the spread of the virus. So let's get started. So first I want to talk about just the coronavirus in general. You've probably seen a lot of pictures of the coronavirus. This is obviously a very simplistic version of it, but I want to just kind of walk you through what I think are the three most important structures to the virus. So first we'll start with this purple part, and this is what's known as the virion envelope. And so the whole point of the virion envelope is it's a protective measure. So just like the skin on your body, it's used to protect what's on the inside. And that's the important part of the virus. And that's what helps the virus replicate. So that's what's known as the single-stranded RNA. So what's interesting about the coronavirus single-stranded RNA is it's known as a positive sense strand. And that's essentially translated to mRNA. And mRNA is so important because that's what gets translated to protein, and that's what our bodies can actually use to replicate further and further. And so I'll talk a little bit more in depth about the single-stranded RNA of coronavirus in a few slides, but for right now, just know that it's mRNA. Next, I'll talk about the spike glycoprotein. So what's interesting about the spike glycoprotein is in our bodies, every cell has a receptor. And what those receptors help with is cellular communication, whether it's through nutrients, chemical signaling, or just straight protein signaling. And so what's unique about these is they're very non-specific. So they can bind to any receptor and essentially get that signals to the cell that they want to get engulfed by the cell. And once they're engulfed by the cell, they can start replicating, and that's when the coronavirus becomes deadly. So these spike proteins are important in order to get the coronavirus inside of our cells. And we'll talk about that in a little bit as well. So this is the general process of how the coronavirus gets inside of our cells and how it can actually start replicating. So I just want to take you through some of these very basic steps, and then we'll go a little bit more in depth on the next slide about how the coronavirus actually replicates. So as we've heard on the news, the coronavirus is a respiratory virus. And so what that means is we either breathe it in through our nose or through our mouth, and then it goes to our lungs, and that's when it can start infecting our body's cells. So this is one of our body's normal cells, and as I said on the last slide, we have these receptors, and these are non-specific receptors. Essentially what it can do is it will find the coronavirus molecule spike proteins, and then those spike proteins, when they bind to those receptors, it signals to the cell and it says, I want you to eat me. So that's what this process is down here. And so this is a very specific process that's called endocytosis. And essentially what that allows the coronavirus to have happen is it allows it to be engulfed inside of what's called a vesicle. And so a vesicle is a way for the cell to engulf some sort of outside foreign particle that it doesn't know what it is, and it allows it to basically break it down to the point where it can figure out what it is. But the coronavirus is smart, and it knows the cell is going to do this. So remember how I talked earlier about how it has that virion envelope structure, and that's used as a protection mechanism. So essentially what happens is in this vesicle, the cell is gonna throw all sorts of chemicals at this virus to try and break it down so the cell can kind of digest it and figure out what it is. But with the coronavirus, it only allows these spike proteins and the viral envelope to break down from the cellular chemicals. And so what that allows is then it allows the mRNA, which is the important part of the virus, to survive in that vesicle. And then what it does is it signals to the nucleus to say, bring this vesicle to the nucleus because that's where the coronavirus wants to replicate. And so we'll talk about that step next. That's why I put a little star here. But essentially, after the coronavirus is able to replicate inside of the nucleus, it creates 1,000 upon thousands of these coronavirus particles that can then be spread to the rest of your cells. So that's the process here. And this is a process either called apoptosis or cell lysis, which means that the cell membrane of our cells break, and that allows all of these coronavirus particles to essentially escape to the rest of the body. And then we start this process all over again and we follow through. And this process happens thousands of times in our body until either our body is able to fight off the virus, or unfortunately the virus would win that battle. And that's when you sometimes see people getting admitted to the hospital and potentially dying from this virus. So next we're going to talk about the replication strategy of the virus. And specifically, we're only going to focus on replication of the virus, but the strategy that's used in the replication is very similar to how our own cells actually replicate our DNA. So we'll start in this box up here, and this is what's known as the central dogma of biology. And so hopefully everyone knows that we are all made up of DNA. And so DNA is basically the genetic code that says this is how our bodies are made, this is how we're going to pass it off for generations to come. So you probably have heard that, the reason you look like your parents is because you have different genes that they passed on to you, and those genes are incorporated into this DNA. And so what the central dogma of biology states is that DNA makes RNA through a process known as transcription, and then RNA can make protein through a process known as translation. And so this is a very important part to my major, genetics, and just biology in general. So next we'll move down to this box. So remember on the very first slide, I talked about how the coronavirus is known as a positive strand mRNA molecule. And so this is a very deadly form of the virus. So just to give you a little snapshot of how deadly this could be, if everyone knows of the polio virus is also a positive strand mRNA virus. So very deadly because it's so easy to replicate within the body, because our body uses positive strand mRNA in order to translate this RNA to protein. And so here, I just have a very basic representation of what the single-stranded RNA molecule could look like. So we have two sides that I want you to know. So we have the 3' end and the 5' end. This is important in any type of mRNA, whether it's viral mRNA or just our body's normal mRNA. And essentially, this tells the body, or any sort of replication strategy that it's going to use, it tells it what direction to read this strand. And so you notice how I have these little shapes in different colors, and these are supposed to represent the different bases that spell out the genetic code. And for this case, it's the genetic code of the virus. So typically in our cells, when you're talking about DNA, there are 4 bases. So there's guanine, cytosine, adenine, and thymine. In RNA, there's also 4 bases. There's adenine, guanine, cytosine, but the different one is uracil. And so in here, basically the way it would work is each of these represent one of those letters, whether, this would be a uracil molecule, this might be a guanine, this might be a cytosine, this might be an adenine, but you'll notice that they're supposed to correctly base pair with one of the other ones. So in mRNA, in any sort of RNA, uracil and adenine will always pair together, and guanine and cytosine will always pair together. In DNA, you would have thymine and adenine pairing together, and then guanine and cytosine pairing together. So just some other points about this. You notice that I have a lot of A's written here. That's known as a poly-A tail. And then you have a big dot here, which is known as a 5' cap. And those are particularly just protection mechanisms for this single-stranded RNA molecule to make sure, no cellular enzymes or no cellular chemicals will actually degrade this RNA. So those just act as protection measures while it's being transported. So now we'll move here and talk about the replication strategy of this virus. So imagine this is a nucleus. So we have the mRNA that came from that vesicle and transported to the nucleus. Then we have these pink blobs, and these are to represent our cell's own replication machinery. And so what makes the coronavirus so deadly is that it doesn't need to use its own replication machinery to replicate. It's able to just use our cell's machinery to replicate. And so since it's a positive strand, in order for it to replicate, what it has to do is it has to put itself into a negative strand. And the negative strand is the process that I show down here. So first what happens is the cellular replication machinery, they find the one positive strand, mRNA, and it starts replicating it in a direction, and it's going to create these negative strand mRNAs. And the reason it has to do this is so then what it can do is it can create 1,000 of different positive strand mRNAs, and those can be used to make other coronavirus molecules. So the positive strand itself can't make a coronavirus molecule, or it can't replicate to make more coronavirus molecules because it'll just replicate into a negative strand. But the negative strand can create a bunch of different positive strand mRNAs, and those are what can be assembled into coronavirus molecules. So that's what I show here. So we have the negative strand that gets selected, and now this one negative strand is going to be used over and over again to create 1,000 different positive strand molecules. And so that's what I'm depicting here, is that we have the cellular replication strategy, and it's going to use that negative strand RNA as a template essentially, and it's going to create 1,000 different of these positive strands. And then we move down here. So then once we have 1,000 of these different positive strand mRNA molecules, that's going to then localize to the cytoplasm of the cell. And in the cytoplasm, that's where the cell is going to assemble a bunch of these different coronavirus molecules. And so once we assemble all of these different coronavirus molecules, we get to this step, and you might remember this step from the last slide. And here is where the coronavirus is ready to spread. It's going to lyse the cell, and then they go through this process again in a different cell. And this is how the coronavirus is so deadly at replicating and becomes so infectious. So I want to take a little break from coronavirus for a minute here and just talk about some of the immune cells that play a role in our body, whether it's to fight against coronavirus or could potentially be used with vaccines to create immunity towards the virus. So I have 4 different types of cells on here, and we'll start with this cell up here, which is known as an antigen presenting cell. So any cell in our body, any foreign bacteria, virus, creates what's known as antigens. And so those antigens are basically little bits of protein that the cell can engulf via endocytosis, and it can figure out what kind of cell is in the body or what kind of cell it needs to go to feed. So an antigen presenting cell, what it does is it processes those different antigens from those cells, and then it will present it on one of its receptors to a different kind of immune cell known as a T cell, which we'll get to in a minute. Next, we have monocytes, and the sole purpose of monocytes is to either engulf debris within the body, so that could mean bacteria or viruses, or it can be used to sometimes induce apoptosis, which is killing of our own cells if that cell might be infected with a virus or with bacteria. So we have a macrophage here, which is basically just like a big stomach. So basically any sort of bacteria or debris that exists in the body, a macrophage is going to go just eat it up. Something is similar with a neutrophil. So a neutrophil is kind of a specialized macrophage that comes into play when the macrophages kind of get too full, we'll say. Then it basically triggers the neutrophils, and the neutrophils are able to engulf bacteria, and they use these little particles that are within the neutrophil, and that's what kills the bacteria. That's not super important, but that's just an interesting point. Then we have a natural killer cell, and the cool thing about what natural killer cells do is they have these receptors, And so remember what I said at the very beginning is that every cell in our body has a receptor of its own. And so what these natural killer cells can do is that they go around to each of our body's own cells and they look at the receptors, and those receptors are either going to say, yes, I'm a good cell, or no, I'm a disease cell. And so for the good cells, the natural killer cells leave them alone and they don't do anything. But if they are disease cells, the natural killer cells can release different chemicals that will actually cause those cells to undergo apoptosis. Next, we'll talk about the T cells. So there's two different kinds of T cells that I want you to know. There's the CD4 T cells and the CD8 T cells. So CD4 T cells are known as helper T cells, and CD8 T cells are known as killer T cells. Helper T cells basically help recruit other immune cells to the area and usually will recruit CD8 T cells to do the killing. Killer T cells is exactly how it sounds. They're used to kill cells that are either diseased or they're used to kill cells that might have a virus in them. And so you'll notice how I have two different colors here, and those are what's known as T cell receptors. And those T cell receptors basically bind with the antigen presenting cell to understand what kind of, it's called differentiation. So this is how our T cells become specialized towards specific type of viruses or bacteria. Once they get the antigen from the antigen presenting cell, they know to change the receptor to be a specific receptor to kill any cell that is expressing that antigen or that bacteria. Next, we have B cells. And so from B cells is how we get our antibodies. And so essentially the way antibodies are produced is we have these big plasma cells. Plasma cells are just antibody factories. They create antibodies once we have exposure to an antigen, virus, or bacteria, and then they release those antibodies, and those antibodies can be used to either kill those antigens by themselves, or what they do is it's called aggregating. So they aggregate around the antigen particle, and then it can get engulfed by a macrophage or a neutrophil. So lastly today, I want to talk about the mRNA vaccine, which you may have seen in the news is what Pfizer is developing currently, and hopefully will get approved by the FDA soon. So first, what is an mRNA vaccine? So mRNA vaccines are really cool because what scientists are able to do is they're able to take mRNA and they're basically able to make it look like a virus. So this mRNA, what it's doing is it's signifying to cells to create what's called a virus-like particle in their cytoplasm. And you'll notice here, this looks a lot like the coronavirus. The only difference is it doesn't have the coronavirus mRNA inside of it. So what this is able to do is it's able to create immunity because our cells now can recognize these spike-like proteins or the envelope, and they can create different antigen immunity towards these. So then when you actually come into contact with a coronavirus particle, your cells will already know, hey, we've seen this before. We need to signal our T cells or our B cells to create antibodies, and then we can start producing those antibodies and the T cells to fight that virus. So I have those listed here. The important cells in mRNA vaccine targeting are the antigen presenting cells, the T cells, and the B cells. So the process of how this vaccine works is we have our mRNA vaccine here. We inject it into people, and then once it's injected into the person, the antigen presenting cell will take that antigen, which is the virus-like particle, and it'll present it to these T cells. So you'll notice here that I kind of split this receptor. So normally it would be a CD4 receptor or a CD8 receptor. And then based on what antigen is being produced, it'll adjust this T cell receptor so they're ready to basically go fight any cell that is presenting that specific antigen. So I kind of break it down here into two different types. So if we have a CD4 cell that's coming into contact with this antigen presenting cell, the CD4 cell is going to go tell B cells, hey, we need to produce antibodies that can recognize this sort of antigen. So the plasma cells then start creating all of these antibodies to which they can release into our bodies. And then you'll notice I have the antibodies here that are released, and they're surrounding a coronavirus particle, to which then gets engulfed by a macrophage, and our bodies don't get infected. Another way this could work is with the CD8 T cells. And so what happens is the CD8 T cell, through this T cell receptor, knows that it has to change its receptor to recognize any cell that might be presenting the coronavirus. So what it does is it goes and it'll find a coronavirus particle, or it'll find a cell that has the coronavirus particles in it, and it releases these chemical particles that can then be used to actually kill the coronavirus or kill those cells. So it's a pretty cool process. These vaccines are very powerful, and I think it's really going to have positive results in the real world. So that concludes our talk today on the coronavirus. I know it was a lot of information. This stuff is very confusing. There are scientists out there that spend their entire careers studying viruses and how to create vaccines for these viruses. So it's definitely not something that you can just learn overnight, but I hope that some of this information maybe makes the coronavirus sound a little less scary, and potentially it can help keep all of us safe, because now you know how to protect yourself. Get the vaccine when it comes out. That'll help a lot. Stay safe, wear a mask, because the coronavirus is a deadly virus, and I hope no one actually catches it. So thank you for all of your time.